And Bats Without Borders is working also in collaboration with other organizations involved in promoting bat conservation, either in Africa, like Bat Conservation Africa, or globally, like their Global Bat Network. So please follow all of them on social media. Okay, so let's start with the seminar. So it's a true pleasure for me to introduce to you today Francisco Amorim, our seminar speaker. I've known Francisco for many years through my many visits to Portugal, and I've also been very privileged to be working with him on one of his chapters of his PhD. So Francisco did his PhD at the Research Centre in Biodiversity and Genetic Resources, also known as CBO, at the University of Porto, Portugal. And his research in his PhD mainly focused on the European free-tailed bat, which is what he's going to talk about today. But generally, he works a lot on the conservation of bats and the interface between applied research and conservation. He's currently doing a postdoc under the project of BatPines, which is promoting the resilience of ecosystem services to climate change using pine plantation, the pine processionary moth, and bats. He's also a committee member of the Cost Action Network on Climate Change and Bats, and he's advising the Portuguese Red Book of Portuguese Mammals. So he's, as you can see, he's quite involved with a lot of conservation, applied conservation research and work. Now today, Francisco is going to be talking about why single species research is important, the European free-tailed bat as a case study. I'm going to stop sharing my screen. And Francisco, I'll open the stage to you.
Okay, thank you, Orly, for the introduction. And let me just share my screen here. It's really great to have so many people from so different places, and I would like to welcome you to this webinar. And I would like to thank, of course, Bats Without Borders for the invitation. So when Rachel first invited me and said we needed a title, I was between two funding applications and a little bit brain dead, to be honest. And I came up with this title, but the truth is I won't actually discuss the benefits of single species research, but instead I will emphasize like the joy of single species research. And how lucky I was actually to be able to devote a substantial part of my research to an amazing species. So if I would have to change my title, I think I would probably go for something like the joy of single species research. That's actually what I'm focusing on. And I will, as Orly said, I will focus on the European free-tailed bat, which was my model system during the PhD. So just briefly, as you know, we live in a world with limited funding, and it's impossible to study each species' ecological process or patterns individually. So sometimes decisions about research priorities can be challenging, and funders normally tend to allocate money towards more community and ecosystem-oriented research. However, funding should actually be targeted projects closing knowledge gaps, regardless of the number of species they are focusing on. And so I like this paper from Lindenmayer a lot, and I'm not actually advocating that single species research is better or worse than community or ecosystem-oriented research, but they should be actually regarded as complementary, each of them with its own strengths and limitations. And well, I suggest to read this paper from Lindenmayer to know a little bit more, but I wanted, for instance, to highlight some important aspects of single species research, as they can elucidate causal process underpinning declines, for example, while also yielding detailed understanding of relationships with the environment that can be applicable to other species in what we know as a model system approach. Of course, it also has lots of limitations too. Often other species are overlooked, and if relationships are very species-specific, generalizations can be quite difficult. Anyway, moving on, I was fortunate to work with lots of people that acknowledge that single species research is also important. And so even before I started my PhD, I started collecting data on one single species. And the interesting part is that I'm not even talking about one of those species that suffer from the cute and cuddling syndrome, or particularly rare or threatened species, which is also an advantage because it can allow us to collect large sample size. So I focused on the European free-tailed bat. For those of you who don't know the European free-tailed bat, it is a species that is listed as a least concern by the IUCN Red List. It has a widespread distribution within the Western Palearctic, and becomes a little bit fragmented. Let me point here. Becomes fragmented in its eastern distribution. And I'm highlighting this issue because I'll get back to this a little bit later. But it is also the only European representativity of the molluscid family, with over 100 species that are mostly restricted to the tropical region. And it's actually one of the only 3 species that reach such northern latitudes. And despite being locally abundant, it was, and I don't know, maybe still is, among the European species with the largest knowledge gap. And this is probably linked to the difficulties, strong and high altitude flyer, and later I will show you just how fast and high they can fly. It is a crevice dweller species with its natural roosts in rocky and cliffy areas, such as the one shown in the pictures. So we knew this species was quite abundant in the northeastern region of Portugal, and particularly in the Sabor River Valley, a valley that was to become flooded in the upcoming years after we started the study, due to hydroelectric infrastructure, as you can see here. So this is already after the flooding. And this dam would lead to the flooding of a total extension of 39 kilometers of the river, and raising the water level from 126 meters to 234 meters. And this would flood an important natural roosting sites for the European free-tailed bat. You can see here, this is a picture of the valley in 2008, so before the flooding, and then you have here in 2016, already after the flooding. I will come to this later, but the flooding was in 2014. And this is a picture from 2016. You can see the flooded area here through all its extension. So given that the valley was going to be flooded, we hypothesized that local bat populations would probably be affected by these drastic landscape changes. And given what we knew about the European free-tailed bat, we thought that this would be a great opportunity not only to study the impact of the dam itself, but also to improve our knowledge on the species. And this was mainly our starting point. But for what we wanted to do and to study the impact, we would need a reasonable sample size. And we still had to sort out how to capture them. We knew from previous studies, including one in north and eastern Portugal too, that Tadaridae species often roost in large concrete bridges. And we had a number of such bridges in our study area. And actually, some of these bridges were over the river valley in the area that was to become flooded, and we had others that were outside the flooded area. These bridges were also used as roosting sites by Tadaridae. You can see here. So they stay in some cracks along the bridges. They roost in some cracks along the bridges. And in some cases, we would discover that we had really high numbers of individuals. So our task was getting a bit easier. We only needed to find a way to capture them. And well, we developed a very simple system consisting of a metal frame that was hanging on the bridges. And then we slided mist nets, as you can see here in the picture. We slided mist nets along these frames, and we were able to pull them up using a pulley system to take the bats out of the nets. And here I know it's a little bit difficult to see, but actually we have two bats here, one and the other here. So this system was extremely effective, and we ended up capturing an incredible number of bats in multiple seasons and years from 2012 to 2017. And this allowed to collect different types of samples, individual information. And we were able to go from diet studies to population genetics and capture marker recapture studies. But eventually, and we were lucky on these two, international researchers also found that our study system was quite nice. So they brought their own ideas and questions, and we studied also communication and flight behavior. So this is the outline of what I'm going to present here in the next slides. I'll start by a more general study on the species evolutionary history, and I will then focus on case studies of how global and local changes affect the species. And finally, I will address more detailed behavioral and ecological aspects, such as diet and flight behavior. Just to let you know that this would never been possible without the collaboration, of course, of all our team members from the Bat Ecology group at CBU, but also lots of all the co-authors, international and national co-authors that made all these studies possible and gave important contributions. So starting for the more general perspective, and what you found about the evolutionary history of the free-tailed bat. So use the standard procedures in phylogeography studies using both mitochondrial and microsatellite markers. In the end, we tried to infer the evolutionary and demographic history using both markers and using approximate Bayesian computation. I'm not going into much details. I just wanted to go back to the distribution map from the IUCN showing the species range to let you know that before we started this study, we had another study by my colleague, Vanessa Mata, where we sequenced the complete mitochondrial genome. And this data set included all the samples that we got from the eastern range of the species. And none of the individuals that we could get samples for from Kyrgyzstan eastwards actually was identified as Tadaria teniota, so the European free-tailed bat. Though this was actually based on only a few samples, it raised the question whether the eastern distribution could actually refer to a different species, and if so, where does the limit of the species range lies? And I would like to take the opportunity to ask if someone has samples from the species, from the eastern range of the species, so from Anatolia eastwards, to please contact me, as we would be really interested to disentangle this story and the real distribution of the species. So moving on, this was one of the reasons why we focused this study on the Western Palearctic, if you want, if we can say so, in the Western range of the species. And in this area, what we found is that mitochondrial markers actually revealed a well-supported split between an European clade, shown here in orange, and then an Anatolian and Middle East clade here more in purple. And when we look at the European clade, we find a second subgroup, but in this case with very, very low support. So we have subgroup B, A, and B for the European range. And looking at the haplotype network, we find a similar pattern, again with the same subgroups, but as you can see, this is differentiating by only one mutation, so very low support. Although what is interesting is that this, what would be subgroup A, is actually restricted to the Iberian Maghreb region, while the other haplogroup, so European haplogroup B, is actually widespread across the range. When you look at the microsatellite data, what we found is that we identified 3 clusters. So a first belonging to the Canary Island, which is completely separated from the mainland Europe. Then we have a second group corresponding to roughly to the European clade, and also showing a slight discontinuity here between what would be group A and group B. And then the third cluster with the Anatolian and Middle East individuals. Though I must point out that we always have a gene flow, so we basically have what is known by isolation by distance pattern within the European range. Of course, the Canary Islands stay isolated. This is probably, which is typical from the islands, probably the result of maybe a recent colonization, because we don't find this pattern in the mitochondrial analysis. So probably a recent colonization event and a subsequent isolation. I won't go into much details here for the evolutionary and demographic scenarios, but I just to show you that we tested different scenarios to get to further conclusions. And basically what we see here is using approximate Bayesian computation, we can test multiple scenarios of colonization and demographic process. Here, for example, it's starting by a colonization of Iberia that then colonized the remaining range. So this is just a quick overview to let you know that we tested different scenarios and hypotheses. And what we did find when we test all the scenarios is that the reconstruction of the evolutionary and demographic history of the species in the Western Palearctic range show us that there was a pre-last glacial maximum colonization from Italy to the Anatolian Middle East. And the species probably used these regions as glacial refugia during the last glacial maximum. And then it colonizes the remaining range, mostly from Italy. And we also find a likely population expansion in the Iberia. So briefly, just wrapping up these results, populations were able to, the Darietanensis populations were able to survive in Italy and the Anatolian Middle East during the last glacial maximum, with the present species range mostly colonized from Italy. And there was a demographic expansion in the Western range. Now, moving on our studies, we go to the implications of global change and the extreme climatic events on species demographic parameters. And here we focused on the effects of severe droughts on the reproductive success of Tadaridae teniotis. So this study used individual data from individuals captured between 2012 and 2013. We assessed also drought severity and biological productivity within the study area. And we used linear models to confirm the effects of these droughts episodes, so the year, over different parameters. Just to show you that we had a severe drought in 2012. As you can see here, this is the precipitation. So this is high precipitation, low precipitation. This would be lower than the long-term median. So in 2012, we have a peak of low precipitation and also normalized difference vegetation index, so NDVI, as a measure of primary productivity. And when we go a little bit further, we can see that this is even more striking during the pre-breeding season. So in the early spring, mostly, late winter, early spring. And the peak, the low peak, it's really striking here. Very low precipitation within this year. And again, primary productivity also really low in this year. So we tried to look what happened to the species during these years. So 2012 and 2013, comparing what would be a normal year, 2013 and 2012. And 2012, the dry year, where we had the major drought, when we look at the female's body condition, we can see it follows an increase from May to October, which is a common pattern for bats in temperate regions. But the interesting part is when we compare to the 2013 results. And you can see there's a huge difference in the early season. So body condition in 2012, the dry year that you can see here, in the early season was much lower than in 2013, the normal year. And what is actually really interesting too is that in 2012, because females, and as we'll see, females have avoided reproduction, as I will show you, they actually had a much steeper increase in body condition until the end of the season, and eventually ending up with higher body condition than in the normal year. But of course, this was with the cost of reproduction. So what happened to reproduction? Oh, sorry, we have another slide here before. So we also saw this, we analyzed the same pattern in males, and you can see that though we have also a lower body condition in males in the early season in 2012, it's not as striking as in females, and eventually they end up with similar body condition. So we wanted to look to reproductive output and see whether this body condition observations were also linked to the reproductive output. And what we found is that we have a much higher proportion of reproductive females in 2013. So you can see here, the black bars represent reproductive females in 2013 and the gray bars in 2012. So in the dry here, we have much less, almost none reproductive females. And when we look at juveniles, number of juveniles captured, we find a similar pattern. So apparently, females avoid reproduction in the 2012, didn't reproduce in 2012, but they invested in maintaining or increasing their body condition to a level that would allow them to survive the next winter. So they avoid the reproductive costs, the reproductive energetic costs of reproduction. Sorry for the repetition of words. So what we found is that droughts affect the reproductive success. We don't know exactly why this happened. It's probably triggered by the low prey availability in the months before the breeding season. Well, of course, the big question is what will happen in arid and semi-arid regions with the predicted increase in drought frequency and severity. So if we have more droughts and if they are more severe, according to the climatic change scenarios, probably bats like Tadaridae teniotis will probably avoid reproduction in multiple years, so decreasing the population. Now, for a more local study, now focusing on the change produced by the dam, actually. So we wanted to analyze what would happen in the roosts that we monitored. Just to refresh your memory, we are at the Sabor River Valley, which was flooded after the dam was built and raising the water level and flooding the cliffs and crevices that are natural roosts for Tadaridae teniotis. As I've shown you before, we had roosts in both within the impact area and outside that area. We focused on these roosts to check for what is known as crowding effects, which represent a specific case of density dependence effects, where a high number of individuals concentrates at available patches after sudden habitat loss. This could be the result of, for instance, of fires or hurricanes. So the principle is that individuals lose most of their habitats or at a local scale or an important part of their habitats, and they will find refuge in the remaining suitable areas. So in this chapter, we used different approaches to test for crowding effects. The first one, we used the capture mark recapture techniques to assess population size and how recapture probability and survival changed over time. And in the second approach, we used the microsatellites to analyze relateness as a proxy for social structure within the roosts. Regarding population estimates over the years, what you can see here is we have the before and after. So this is 2014, after the flooding of the river, so after the flooding of the dam. And we can see clearly that in 2015, so immediately after the flooding, we found that the population estimates peaked at safe roosts in the impact area and also in the control area here, but to a much lesser extent. So we had a higher increase of population estimates here in the impact area, with many individuals probably arriving to this roost, the safe areas. But in the second year after flooding, estimates actually returned to similar estimates of population size. So this seems to be a transitory effect mostly. And what is interesting too is that when we look at recapture probability, here represented here in the different years, so this is always between years, so this would be from 2012 to 2013 and so on. And you can see that after the peak, so in 2015, we have a decrease in the recapture probability. So this is probably linked also to the arrival of new individuals in 2015, which then travel or go to other areas that also, or else they don't stay in the roost. Actually, they don't stay in the roost, they leave the roost. So we find this lower recapture probability between the first and second year after flooding, as I was saying, probably due to the movement of individuals to the safe roosts and then going away from these roosts. And although we did not find any effects on the apparent survival of the individuals, we find some signs of what is called density-dependent effects on apparent survival. And well, when we have high populations, probably survival will decrease. But as you can see, the confidence limits are really high. So the confidence limits are really high. So it's not very clear what we have here. When you look at relatedness, and this is a little bit more, I'll try to guide you because the plots are not very brilliant, and actually we are trying to improve them a little bit. I'll try to guide you through. They represent the relatedness difference between a given year and all the remaining years at the bridge level. So we are comparing relatedness within 1 year to all the others, but always at the bridge level. So when we see here the dashed lines, here it's what we would expect if they were they were arranged by chance. The orange line is the cumulative of this difference. So for if they were arranged by chance, and the strong black line is what we actually observe. And normally, what we see in the different years before the impact, at the control area or before the impact, is that the observed value lies either within the confidence limits or a little bit to the right, meaning higher relatedness within a year comparing to the all remaining years. But when we see this after the flooding, the first year after the flooding, we see an impressive decrease on the relatedness at the impact area, but not followed into control area. So this is once again probably due to the arrival of new individuals to the roost that we are monitoring at the impact area, which are probably coming from many different roosts, and this led to a decrease in the observed relatedness. But then again, on the second year after flooding, we find the expected pattern again. So it seems that indeed we have crowding effects after flooding, with population size increased at safe roosts. And apparently this does not affect survival, the flooding itself, but there's a density-dependent effect on survival on the other hand. And well, this is also accompanied by a decrease in relatedness at the refuge in the impact area, probably due to the arrival of new individuals. Overall, I must say that this is mostly transitory. So we don't know actually where the individuals are going after the second year, but they moved from the roost that we were monitoring apparently, and we don't know what is happening probably in other colonies or if they occupied other roosts that were unoccupied. Moving on and narrowing in a bit down to behavioral aspects, I'll start you to show a study that was led by my colleague, Vanessa Mata, and that focused on the diet and the sexual segregation in diet composition. So here we use fecal pellets, individual fecal pellets from 143 individuals collected between April and October and in different years. We then used the metavar coding approach, using first a PCR to amplify a CO1 fragment, and then next generation sequencing to allow us to get sequences for the different species. And with the bioinformatics, we're able to actually identify some of the species. I won't say most, but some of the species at least. And then we compared what we find about the species ID to sex and age and seasonal difference using generalized linear mixed models. Well, so regarding general aspects of the diet, we found that Tadarida is eating mostly Lepidoptera, which is not a surprise. Of course, most studies already pointed to this, but what was amazing was actually the resolution that we were able to get afterwards. So we got 17 families and then 180 species. And we wanted to move a little bit for the species analysis. So we wanted to check what was happening in terms of sex. And we analyzed the difference between the diet composition in both males and females. And we did find difference in the diet composition. And when we look at the females, what we are seeing is that they are mostly eating bigger moths and migratory moths, while males are actually eating smaller moths, and most of them are non-migratory moths. And we aim to test this idea, and we actually find that, can see here, is that they are selecting different prey traits. So in terms of size, both in terms of size as expressed here by wingspan and the migratory behavior. And females, as we showed before, are actually preying on bigger migratory moths and bigger moths, bigger preys, and most of them are actually migratory preys. So this study highlights resource segregation between females and males, which is probably related to the higher energetic demands of females during the pregnancy and lactation, so during breeding season. And the results also suggest that females may be more susceptible to global changes driving moth migrations, while males may be more vulnerable to local prey declines related, for instance, to land use changes, since the ones are eating more migratory moths and the others are eating more non-migratory, so local species. So moving on to the last case study, this was the work led by Tigo Mario, many of you will know, and has already presented the Winged Wednesday webinar too. And this was a really amazing opportunity, I must say. In this study, we used GPS loggers, and we were able to retrieve data from 8 tags from the 33 that we deployed. And by linking this GPS data with high resolution to wind and digital elevation models, we were then able to characterize the flight behavior of the species, and also we were able, using generalized additive models, to predict where high altitude flights are likely to occur. So I'm just giving, although these were not the goal of the study, I'm giving just these bits of information that I know people normally like. Here you can see the tracks from the different individuals. And what we did find is that most individuals emerged around 45 minutes after sunset, and they flew constantly through the night without stopping. They either returned to the roost, or they could stop at roosts that were 65 kilometers apart. And the maximum distance traveled by an individual was 184 kilometers in one single night. So when we look into speeds, to their speeds, and particularly to hair speeds, I can explain this later if you want to ask. We can see that they mostly flew at low speeds, so what would be predicted according to their size and wingspan and body mass, but occasionally they reach really, really high speeds, unexpected for the species traits. So this is hair speeds, and we had speeds of over 130 kilometers per hour. When we look to their flight altitude, you can see here is the representation of the terrain in this black line. And when we add the tracks, you can see the altitudes that these bats are covering. So they mostly follow the terrain, as you can see here, but occasionally, from time to time, they will go really high, these high ascents that are normally associated to high slopey areas and mountain areas. You can see here another representation of the track, so a 3D representation. And you can see that these ascents normally occur actually where we have higher slopes. And we hypothesized that bats were actually taking advantage of orographic uplift until the wind support drops. And what is this about uplift? Just to explain you the idea is that when wind hits rugged terrain, so you can see this is the wind direction, and we have the mountain areas, then it will create a vertical component of wind, and bats will take this chance to go upper. And then when they don't have this support, they will come down again. And this is the same thing that you can see here for one of the bats that we study. So this is orange lines are the wind, the vertical wind component. If they are larger, they mean that it's more windy, so it's strong. The vertical component is stronger, is higher. And you can see this is the starting point of the track and the finish point of the track. And you can see they are flying and taking advantage of this wind on the landscape to reach these really high altitudes that go to 1,700 meters. And then when they don't find that much support from the wind, they will come down again. And what was really interesting is that when you consider both the orographic and the wind components, the vertical wind components, we are actually able to predict where these high accents are most likely to occur. And this is what we've done for the study area, portion of the study area, because this is really complex given the detail, the 3D detail and everything. And you can see here the red colors represent where high accents are likely to occur. So mostly in the valleys areas where they also have high wind support. So in this study, we found that European free-tailed bats use uplifting winds to ascend to over 1,600 meters above ground level. These high elevation accents are predicted by geography with orographic uplift. And we found that these bats can fly at self-powered airspeeds over 130 kilometers, and they can also explore the energy of the landscape similar to diurnal birds. Of course, it's different because we know that bats take advantage of the thermal lifts, and the bats are not doing this because they don't have thermals during the night, of course. So I just wanted to tell for this specific work that this was actually privileged to work with all these people, and we had lots of fun during this work. It was an amazing opportunity, and I really like this study. Well, I like them all, but this one in particular, I think it's really interesting. So will we have more on the European free-tailed bat? Yeah, I think so. So without taking much more of your time, I wanted to say that all the knowledge that we gathered also allowed us to write a chapter for the handbooks of the the mammals of Europe. We wrote a chapter on the European free-tailed bat, which should come out briefly, I suppose. And we expect to have more research in the upcoming years. And you can follow, if you want, you can see our project on ResearchGate. We have a project that gathers not only our research, but all the research, or at least we try to keep up to date with most of the research, at least on the European free-tailed bats. And we still have more to come. Just to tell you a little bit, we have this collaboration with Kirsten Bohn from the John Hopkins University. We are looking at communication in the species. So we have assembled SM. This one was the first SM song meters automatic recorders. So we have assembled them, and we have lots of data to analyze for the communication, species communication. We have done some steps, but this is still ongoing. Recently, my colleague Vanessa Mata also got a British ecological grant to study a little bit more in detail the diet of the species. So we are now going for over 1,500 individual fecal samples, and we want to test different the difference in phenological periods, the difference between reproductive females and non-reproductive females, and also difference between season and between years. And we'll be doing this also with Tiago Pinto, which is a master student working with us now. And well, in the end, I like to believe that we are making significant progress towards species knowledge, and we hope that this is also relevant for other species. And well, I hope I somehow convinced that single species research is also important and can actually pay off. And of course, we still have lots of questions. And I think the more studies we have, probably the more questions we'll have too. And so that's it. And well, thank you for your time. And I'm happy to answer your questions now.
Please join me with thanking Francisco for a great talk, and great to hear so much research carried out on such an incredible species. Who would have known bats can actually outperform birds on some levels?
True.
So I'm waiting, so if anyone has any questions, please add them to the chat. But while we're waiting for people to add their questions, let me ask you a bit of quite a general question, I suppose. So you studied a lot about the ecology of this species, and in fact it's an interesting species in the extent that it flies such far distances. The diet, in a way, it doesn't seem like they're that constrained by competition or anything like this, because they can exploit all these high elevations. I wonder, what do you think is the true, in a way, conservation threat for these kind of species that are so widely dispersing?
Well, great question. So that was one of the starting points, actually. It's a widespread species, and the population seems to be really, really big. Also, at least they are locally abundant where they have conditions. But as you saw also, they have some specific threats, and we've seen so it's a species, in this particular case, it's highly associated to the Mediterranean area, which is an arid area. And you saw that, as I show you, so we have a drastic decline, for example, on the reproductive success when we had a drought here, and with climatic changes, for instance, we expect that this will happen much more often. And if we put this on top with the local changes, as I also show with the dam, so with the crowding effects and all that kind of aspects, actually we could be overlapping multiple threats to one species that is abundant, true, but there are some case studies also with other abundant species that suddenly disappear. I don't remember exactly. It's a bird species in the UK, maybe you know, that went almost extinct, and it was really abundant in the last year. So the main point here is that global changes and local changes together can actually threat really abundant species too, I think.
Yeah, great. And that's a really important message, actually, in terms of conservation, where we focus.
Yeah, and the thing is that normally we focus on the more threatened species, and we disregard these common and abundant species.
Yeah, indeed. So we got some questions coming up. I'm going to start with a question from Marie Handley, asking, how did you know how high the bats flew over the mountains?
Maybe that was not clear. Okay, so maybe I'll just go back. I think it's here. So we use GPS tags, and the GPS tags, they also measure altitude. So okay, so you can see the representation. So we have the altitudes of at which the bats are flying, and then because we also have the elevation models, we also have the altitude of the terrain. So basically, by using the difference between the GPS tags, the altitude of the GPS tags, and taking out the elevation of the terrain, we know how high above the ground they are actually flying. So this is, for instance, here we can see this is 2,000 meters above sea level, but it's actually would be actually around 1,600 meters, because we have to take out the terrain elevation. So there's no, how would I say, that's not like some sort of magic. It's just taking out the altitude of the GPS tags and subtracting the elevation of the terrain. So we know the GPS position, also the lat long position, and we just need to take out the elevation of the terrain. But of course, this was only possible because the GPS tags that we were using had high resolution. We were taking the location at every 30 seconds, so we can easily associate to the terrain.
Oh, it's amazing the advances in tracking technology now that you can use GPS tags on bats. We have another question from Quincy Augustine asking, seeing that this species has such a wide range, do neighboring countries have similar conservation strategies?
If they have similar conservation strategies. So this is again a problem with the widespread species. So they don't have really target conservation. So this is we are not talking about any conservation guidelines here. We know what could be affecting the species, or at least we have some ideas now. But we don't have, even in Portugal, we don't have specific guidelines for the conservation of widespread species and locally abundant species. So that's the problem, and links to the first question by Orly. It's like these species are often disregarded in terms of conservation.
Thank you. And we got a question also from Javier Juste saying, great talk, Francisco, and asking how much trophic niche partitioning is affected or explained by sexual dimorphism?
Interesting question. Very interesting question. I don't know the answer to that, but at least in Portugal, we don't find in this species, we don't find such huge difference between females and males. So we don't have sexual dimorphism, at least from what we know so far. Maybe Javier knows a bit more and can add to this question, I'm sure, but at least from the individuals that we were capturing, we didn't find sexual dimorphism.
Okay, thank you.
Not sure if Javier wants to add anything else to this topic.
Javier, if you want to turn your microphone on, if you want to say anything about.
Yeah, well, Francisco, congratulations. That's a great talk. No, I was wondering, because this important sexual dimorphism in molluscs in general, this sexual selection going on in this group, particularly of bats, and I know that Tadarida tinnitus is not very clear, but still in the mouth, particularly in the teeth, the male are much bigger than females, and so I was thinking that this could affect to the diet.
The mouse, you say?
Yeah, the males are normally bigger and stronger, and they have a stronger mouth, and so I thought that this could be affecting to this the results you were.
The males, you say?
Yeah.
But then I would predict that they would eat probably the larger moths, right? And that's what's happening is exactly the opposite.
Yeah, or at least they can capture from more long distance moths, because they probably of the ecolocation is also a little bit lower because they are bigger, and there's probably there would be a mixture of things that are affecting to the diet. This interesting finding, not only the energetic demands that they could, which is also clearly different.
Okay.
between females and males.
Okay, well, I think that's really interesting. It's something that we didn't look at actually, and we could probably, may or maybe, but I think, well, that the thing is that females during pregnancy and lactation, they have high energetic demands, so they really need to feed to take the most out of their food. So they probably take the opportunity of feeding on migratory moths and larger moths, because with less effort they can have actually much more calories and
that's
and that actually, if males don't have the parental cares and everything, I think this will also contribute to the success of the species, right? Because the survival of juveniles, for instance, is it's totally dependent on the females. So maybe this is also a question of species survival. I'm not sure. I'm just making this up a little bit.
That is probably true. Yeah, thank you.
Thank you.
But it's interesting, this interplay between reproductive needs versus morphological size, which sometimes may be the results, for example, with male-male competitions for mates. So
yeah. Maybe with this new sample that we'll have with much more detail, with 1,500 individuals, maybe we can look to much more detail what is happening. We hope. So we hope.
I look forward to hearing more about this. If anyone has any last questions, please share them in the chat or raise your hand. I'll just mention that Lee Richard said, fascinating talk. Thank you for sharing, Francisco.
Thank you.
So I'm going to give people a few more seconds if anyone has any other questions they want to ask. But in the meanwhile, while we're waiting, perhaps I can ask you a question that relates more to my personal interest in patterns of gene flow across the landscape. Just wondering if you think that there is anything that would affect connectivity in terms of functional connectivity, so gene flow in these kind of species, or do you think that indeed across Europe we can regard it as an admixture population, a single population across the whole of Europe?
That's a really, really good question. I don't know. Maybe if we could look now to SNPs or new markers, we could find the different thing, or maybe nuclear markers or whatever. But at least from what we know, and considering that the species can fly such long distance in one single night, I think it could be it's completely admixtured, and we could have like a sort of a single population that is only isolated by distance, as I show. I think it's reasonable, but maybe with higher resolution we could disentangle that.
Yeah, excellent. Well, we are right here on time. It's 3 o'clock or 4 o'clock in Central African time. So please, everyone, join me with thanking Francisco for a really, really great talk on a really interesting species. Right. Well, thank you very much,
Perfect.
Francisco.
Perfect. I really enjoyed. Thank you for the invitation. I hope you liked too.